Hello and welcome to this learning module. Today we will provide an overview of the fluid balance in the human body. This video is intended to supplement any readings or coursework assigned by your instructor. The objectives for this learning module are: explain the concept of fluid balance, including the purpose and mechanisms for maintaining homeostasis, identify the major fluid compartments of the body, and define the terms isotonic, hypotonic, and hypertonic. Human beings are mostly water, ranging from about 75% of body mass in infants to about 50 to 60% in adults to as low as 45% in old age. Therefore, fluid balance is key to maintaining the body's equilibrium that is necessary for optimal functioning, a term referred to as homeostasis. This system ensures the body's internal environment remains stable, allowing cells, tissue, and organs to perform their functions efficiently. Adequate hydration and precise regulation of fluid levels are essential for numerous physiological processes, including temperature control, nutrient transportation, waste elimination, and the maintenance of blood pressure. The body controls this balance through mechanisms like thirst sensations and the filtration capabilities of the kidneys. Disruption in fluid balance can have significant repercussions, leading to conditions like dehydration or electrolyte imbalances. Fluid compartments are locations that are largely separated by some form of a physical barrier. The intracellular fluid compartment is the system that includes all fluid enclosed in cells by their plasma membranes. Extracellular fluid surrounds all cells of the body. Extracellular fluid has two primary constituents: blood plasma and the interstitial fluid that surrounds all cells not in the blood. These compartments are illustrated in the image on the left. The intracellular fluid lies within the cells and is the principal component of the cytoplasm. As we see in the image on the right, the intracellular fluid makes up about 60% of the total water in the human body. The extracellular fluid accounts for the approximate other one-third of the body's water content. An estimated 20% of the extracellular fluid is found in plasma. Plasma travels through the body in blood vessels and transports a range of materials, including blood cells, proteins, including clotting factors and antibodies, electrolytes, nutrients, gases, and waste. On average, a person has about 11 liters of interstitial fluid that provide the cells of the body with nutrients and a means for waste removal. Gases, nutrients, and waste materials travel between capillaries and cells through the interstitial fluid. Cells are separated from the interstitial fluid by a selectively permeable cell membrane that helps to regulate the passage of materials between the interstitial fluid and the interior of the cell. The body also has other water-based extracellular fluid. These include the cerebrospinal fluid that bathes the brain and spinal cord, lymph, the pleural fluid in the pleural cavities, and the peritoneal fluid of the peritoneal cavity. Preserving the delicate balance among the fluid compartments of the body is essential to maintain homeostasis. The fluids of the body exist as a solution of dissolved elements and water. Cell membranes are semi-permeable, meaning they allow lipid-soluble substances to move into and out of the cell freely, while not allowing water-soluble substances to cross the membrane. But to sustain life, both types of substances must be allowed to enter and exit the cell. Materials will flow from an area of higher concentration to a lower concentration, which creates a concentration gradient. Some compounds can travel freely across the cell membranes and require little to no energy or ATP. Others require active transport, which uses ATP because of the size of the compound or because of an incompatible electrical charge. Diffusion, filtration, and osmosis are cellular transport mechanisms that help preserve fluid balance. Diffusion is the movement of molecules from an area of high concentration to an area of lower concentration. We have already established that passive transport, illustrated in the image on the left, does not require energy or ATP. Some examples of substances that use this process are oxygen and carbon dioxide diffusion between the alveoli of the lungs and the pulmonary capillaries. Active transport allows cells to move a substance against its concentration gradient through a membrane protein and using ATP. For example, the sodium-potassium pump employs active transport to pump sodium out of the cells and potassium into the cells, with both substances moving against their concentration gradients, as illustrated in the image on the right. Filtration uses a hydrostatic pressure gradient that pushes the fluid and the solutes within it from a higher pressure area to an area of lower pressure. Filtration is an extremely important process in the body. For example, the circulatory system uses filtration to move plasma and substances across the endothelial lining of capillaries and into surrounding tissue, supplying cells with their nutrients. Oncotic pressure, also called colloid osmotic pressure, is the force that pulls fluid back into the capillary. It has the opposing effect of hydrostatic pressure. In capillaries, hydrostatic pressure is higher than the oncotic pressure at the arteriolar end of the capillary. This pressure forces plasma and nutrients out of the capillaries and into surrounding tissue. Fluid and cellular waste in the tissue enter the capillaries at the venule end, where the hydrostatic pressure is less than the oncotic pressure in the vessel. This is illustrated in the image seen here. Hydrostatic pressure is especially important in governing the movement of water in the nephrons of the kidneys to ensure proper filtering of the blood to form urine. In the body, water moves between compartments through semi-permeable membranes from one compartment of the body to another by a process called osmosis. Osmosis is basically the diffusion of water from regions of higher concentration to regions of lower concentration. As a result, water will move into and out of cells and tissue depending on the relative concentrations of the water and solutes found there. An appropriate balance of solutes inside and outside of the cells must be maintained to ensure normal function. Two solutions that have the same concentration of solutes are said to be isotonic. When cells and their extracellular environments are isotonic, the concentration of water molecules is the same inside and outside the cells, and the cells maintain their normal shape and function. A critical aspect of homeostasis in living things is to create an internal environment in which all the body's cells are in an isotonic solution. Osmosis occurs when there's an imbalance of solutes outside of a cell versus inside the cell. A solution that has a higher concentration of solutes than another solution is said to be hypertonic, and water molecules tend to diffuse into a hypertonic solution to dilute it. Cells in a hypertonic solution will shrivel as water leaves the cell via osmosis. Conversely, a solution that has a lower concentration of solutes than any other solution is said to be hypotonic, and water molecules will diffuse out of a hypotonic solution. Cells in a hypotonic solution will take on too much water and will swell and burst. Let's take the concepts of isotonic, hypotonic, and hypertonic, which are collectively referred to as tonicity, a step further. This concept becomes extremely important when choosing IV fluid that will best suit your patient. The two most common pre-hospital isotonic solutions are 0.9% normal saline and lactated Ringer's solutions. Because the concentration of these IV fluids is comparable to blood, the fluid stays in the intravascular space, and osmosis does not cause fluid movement between compartments. Isotonic solutions may be used to raise the blood pressure in patients who are hypovolemic. These patients must be monitored closely to ensure that we do not overload them with fluid, which can lead to serious complications. When hypotonic IV solutions are infused, it results in a decreased concentration of dissolved solutes in the blood as compared to the intracellular space. This imbalance causes osmotic movement of water from the intravascular compartment and into the intracellular space. For this reason, hypotonic fluids are used to treat cellular dehydration. An example of a hypotonic solution is 5% dextrose in water, or D5W. D5W is a unique fluid because while in the IV bag, D5W is considered an isotonic solution, but once administered, the dextrose is quickly metabolized in the body and the solution becomes hypotonic. The primary use for D5W is when mixing medications for IV infusion. Hypotonic solutions can cause a sudden shift of fluid from the intravascular space to the cells, leading to cardiovascular collapse and increased intracranial pressure. It's for this reason that we do not use D5W in patients suspected to be experiencing a stroke or who have sustained head trauma. Clinicians use hypertonic fluids to increase intravascular fluid volume. An example of a hypertonic IV solution is 3% sodium chloride, also called hypertonic saline. When infused, hypertonic fluids cause an increased concentration of dissolved solutes in the intravascular space when compared to the cells. This causes the osmotic movement of water out of the cells and into the intravascular space to dilute the solutes in the blood. This increase in intravascular volume increases mean arterial pressure, stroke volume, and cardiac output when compared with equal volumes of normal saline or other isotonic fluids. When administering hypertonic fluids, it is essential to monitor for signs of hypervolemia, such as breathing difficulty and elevated blood pressure. This brings us to the end of this presentation. Here we see the learning objectives for this module. Can you demonstrate each of these objectives? If not, consider going back to review any content that you struggled with. Thank you for watching this learning module.